Okay, good afternoon, everybody. It's another bright afternoon that I would like us to look at the nervous system and the study of the structure and organization of the nervous system is what we refer to as the neuroanatomy. So we want to look at neuroanatomy to see basically the structure, organization of the nervous system. Nervous system is a very, very wide, detailed system that we might not be able to finish in one presentation, but we shall try to address the most important aspects of it that we require to understand. So the simple definition of anatomy is basically the study of the body structures, and therefore the neuroanatomy itself is a branch of anatomy, and it deals with the study of the structure and organization of the nervous system. That is the basic definition of those terminologies. So what is the nervous system? We are saying that it's a rapid communication system. You need to remember in the body we have two communication systems, and that is the nervous system. The other one is the endocrine system. So the nervous system, being a rapid communication system, it simply receives chemical signals and it provides information about changes in both internal and external environment. So it is a communication system communicating between the external and internal environment. So it provides equilibrium. For example, if I talk of temperature, the normal range of temperature we know is 36.5 to 37.5. So it is supposed to be maintained within this range. Anything below this range, the nervous system will sense. Anything above this range, the nervous system will also detect, and therefore it will inform the relevant effector organs to act in order to bring it into the normal range. So what is the classification of the nervous system? You have two major classifications of the nervous system, and that is the central nervous system and the peripheral nervous system. The central nervous system consists of the brain and the spinal cord. The peripheral nervous system here consists of the somatic nervous system and the autonomic nervous system. The somatic nervous system has got 12 pairs of cranial nerves and 31 pairs of the spinal nerves. The divisions of autonomic nervous system include the sympathetic nervous system and parasympathetic nervous system. Sometimes we also include the enteric nervous system. So at least there are some various issues that we need to know about the brain, the spinal cord, the cranial nerves and the spinal nerves, and also the autonomic nervous system. This is still the classification, but this time round we have included just the parts of the brain, what we are calling the forebrain, the midbrain, and the hindbrain. Some important aspects that you need to know about the brain and also the spinal, the cranial and the spinal nerves is that the weight of the brain is about 1.3 to 1.4 kilograms. So the human brain weighs about 1.3 to 1.4 kilograms. In the human body, we have 12 pairs of cranial nerves. These 12 pairs, 4 of them are mixed and 3 are sensory, while 5 of them are pure motor nerves. When we talk of mixed, we mean that is a nerve that consists a sensory component and a motor component. So how do you tell which one is mixed, which one is motor, which one is pure sensory? Sometimes we use some kind of statements that can help one to remember. For example, if we can just come up with a statement, let's start by writing the numbers first. That is number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So, if we coil around a statement and say maybe, a statement like this, some say money matters, but my brother says big brain matters most. Some say money matters, but my brother says big brain matters most. So if we take S here, then it means the cranial nerve number 1 is a sensory nerve. When we take another S here for the second word, it means also the second cranial nerve is pure sensory. When we take M from number 3, it means cranial nerve number 3 is pure motor. When we take M from number 4, it means the cranial nerve number 4 is pure motor. When we take a B from number 5, B means both. That is both sensory and motor. Sometimes can be mixed. So both is the same as mixed. So cranial nerve number 5, it has both motor and sensory component. Cranial nerve number 6 is purely motor. Cranial nerve number 7 is both. Cranial nerve number 8 is sensory. Cranial nerve number 9 is both. Cranial nerve number 10 is both. Cranial nerve number 11 is motor. And cranial nerve number 12 is actually motor. So that's just some form of shortcut ways of remembering which of the cranial nerves is motor, which one is sensory, which one is mixed, and so on and so forth. So after knowing something on the cranial nerves, you also need to know that there are 31 pairs of the spinal nerves all of them are mixed. They contain both motor and sensory. The length of the spinal cord is 42 to 45 centimeters thereabout. And we are saying that in adults, the spinal cord terminates at L1, L2. That is lumbar vertebral 1 and lumbar vertebral 2. So the clinical importance of this region is that when it comes to anesthesists, when they want to do a lumbar puncture, they normally locate L3, L4. Remember, our spinal cord has ended at L1, L2. So the anesthetists are pretty sure that when you locate L3, L4, then you are very sure that you will not injure the spinal cord, because the spinal cord will be up there. That is the relevance of this aspect. There is also something we call cerebrospinal fluid, what we refer to as the CSF. The volume of CSF is about 125 mils to 150 mils. The significance of CSF volume is that whenever there is a pathology in terms of production and flow, for example, if its flow is obstructed, then we end up getting a condition known as hydrocephalus. We also have an aspect of the coverings of the brain. We have connective tissues that cover the brain. We shall see in the subsequent slides. We call them meninges. And we shall also see the issues of hematoma whenever we shall be looking at that aspect. Lumbar puncture we have already mentioned. And then we have something called upper and lower motor neuron lesions. When we will be looking at sensory and motor pathways, we shall be able to mention something about the upper and lower motor neuron lesions. So the human brain looks like this. This is how the human brain looks like. And there are three main parts. What you are seeing, the large portion is what we refer to as the cerebrum. There's a small portion down here. Down here, we call it cerebellum. And then what is projecting toward the spinal cord is what we refer to as the brainstem. In 1909, thereabout, just a little bit of history of neuroanatomy, there was a German anatomist by the name Corbinian Brodmann. So this is a German neuroanatomist who was born in the year around 1868. He tried to analyze the brain in terms of specific areas and their functions, and he mapped the whole cortex of the brain in terms of numbers and their function. So every area he gave it a number, and he also gave it his name. For example, the area that deals with the sensory aspects, that is interpretation of the sensory stimuli, is area number 1, 2, and 3. This is this region here, 1, 2, and 3. So these scientists named these areas as Brodmann Area 1, Brodmann Area 2, and Brodmann Area 3. The area that deals with the motor aspects, he classified that area as Brodmann Area 4. The area that deals with the visual interpretation, he classified it as Brodmann Area 17, that is on the occipital lobe of the brain. So this area, Brodmann 22, is the Wernicke's area that deals with language comprehension, and so on and so forth. So he mapped the brain into 52, total 52 areas. So this guy, Corbini and Brodmann, that is the work he did in around 1909, after having been born in 1968, not 1868. After doing this great work, about 1909, 9 years later, he died at the age of 49. So this scientist, that is what we can mention about him. This is just a summary of some of the areas that Brodmann classified, and you can see areas 1, 2, 3, they deal with primary somatosensory interpretations. Brodmann Area 17 is a primary visual cortex, and so on and so forth. What you are seeing here is the brain, and this flap you see down here is the meninges. So your brain is covered by 3 connective tissues that we refer to as meninges. So from outside towards the inside, we have the dura mater. Then we have the arachnoid mater, arachnoid. Then we have the pia mater. So we can say there are 3 connective tissues that actually cover the brain, and these tissues are what we are referring to as the meninges. The most important aspect of these meninges is when it comes to head injury, that is the clinical correlates, and any bleeding that occurs below, actually let's start with above the dura mater is what we refer to as epidural hematoma. Any bleeding that will occur below the dura mater, then we call it subdural hematoma. You can also have bleeding in the arachnoid, below the arachnoid region, and that is what we refer to as subarachnoid bleeding. We shall see in the subsequent slides. So basically, this is the organization of the head, that is from the skull all the way to the cerebral cortex. So what you are seeing in red is the skin on top of the skull, which we refer to as scalp, S-C-L-P. So from the scalp, which is the skin on the head, we go now to the bone itself, and the bone, what you are seeing in yellow, is what we refer to as the skull. So what is in yellow here is the skull. Then we have the meninges. What you are seeing in blue is the dura mater. The portion of the dura mater that touches the skull is called periosteal dura mater. And then the aspect or the portion that is in direct contact with the brain, that is in direct contact with the arachnoid mater is what we call the meningeal dura mater. So you can see the dura mater has two layers in itself, a layer that is in direct contact with the skull, and the lower layer that is in direct contact with the arachnoid matter. So when there is a bleeding between the upper layer of the dura mater and the skull, we call it epidural hematoma. Where there is a bleeding below the dura mater, that is between the dura mater and the arachnoid mater, we call it subdural hematoma. Any bleeding below the arachnoid mater, we call it subarachnoid hemorrhage. You can also have meningeal hemorrhage within the cerebral cortex. Those are just some of the findings on the CT scan of the head when a patient gets what we refer to as head injury. So this one explains what I've just mentioned. There is a bleeding here above the dura mater, and this is what we refer to as epidural hematoma. There's another bleeding below the dura mater, what you are seeing in blue, and that is what we are referring to as the subdural hematoma. So, epidural hematoma occurs when there is rupture of a blood vessel called the middle meningeal artery, while the subdural hematoma occurs when there is rupture of cerebral veins. So mark those two differences. Subarachnoid hemorrhage, we have already mentioned it, is actually the bleeding below the arachnoid mater. It is also mostly caused by a rupture of an aneurysm. Aneurysm is just the bulging of the blood arteries in the brain, particularly at the base of the brain. So when it ruptures, a patient gets what we call subarachnoid hemorrhage. When you want to diagnose subarachnoid hemorrhage, if you do a lumbar puncture and analyze CSF, then you are likely to get blood within that sample. So we have talked about we mentioned something about the cerebrospinal fluid. This is a fluid that is found within the central nervous system, that is the brain and the spinal cord. So, CSF, if I may just draw an illustration here, we want to talk about production and drainage of CSF. So let me just make a sketch here to show what I want us to talk about. So these are the ventricles. This is what we call lateral ventricle. This one is the third ventricle. This one is the fourth ventricle. And this one down here, there's something called central canal here. It's just going down into what we refer to as the spinal cord. So what you need to know on production and drainage of CSF, that is cerebrospinal fluid, is that within the brain, there are spaces we refer to as ventricles. And a ventricle is just a space. And also, there's another terminology you need to know, the foramen. Foramen. The word is foramen. Foramen is just an opening, while the ventricle is a space within an organ. The two organs that we normally get ventricles is actually the heart and also the brain. So in the brain we have ventricles, those are spaces. And those ventricles are these ones here. The two ones you are seeing up there, we call them lateral ventricles. Then we have the one in the middle, what we refer to as the third ventricle. And then we have the last one, which is the fourth ventricle. So our CSF is produced in the lateral ventricles from what we call choroidal plexus. Choroidal plexus. Choroidal plexus are just a network of capillaries within the brain, particularly in these areas. So from ultrafiltration, we get our CSF in the lateral ventricles. So from the lateral ventricles, our CSF will flow to the third ventricle through this foramen here. So we are saying from this diagram, though it's not very clear, CSF will be produced from the lateral ventricles, particularly in what we are referring to as the anterior horn. And then what we are seeing here is the third ventricle. And what you are seeing down here is actually the fourth ventricle. So our CSF will come all the way from the lateral ventricles, and it will get into the third ventricle through a foramen that we call the foramen of Monro. Foramen of Monro. So this is the foramen of Monro. That is from the lateral ventricles to the third ventricle. From the third ventricle, our CSF will flow into the fourth ventricle through another foramen you see here. We call it aqueduct of Sylvius. Aqueduct of Sylvius. One clinical importance of this foramen here is that it is very narrow and it is the commonest site of blockage obstructing the flow of CSF from the third ventricle to the fourth ventricle, therefore contributing to a condition we refer to as hydrocephalus. Hydrocephalus. This is a condition where there is excessive accumulation of CSF in the brain. Remember we said the normal range of CSF, the amount, is about 125 mils to around 150 mils. So anything above that, you get this condition called hydrocephalus, meaning there is obstruction. So whenever there is obstruction or excessive production of CSF, then we end up getting this condition here. So that marks the end of the production and drainage of CSF. It was just a summary as you have seen. What you need to remember just before we move there is that once CSF has reached the fourth ventricle, it has two options. It can get into the subarachnoid space, that is the reabsorption by substances or the villi, subarachnoid villi. So CSF can be reabsorbed back through what we refer to as the foramen of Luschka. that is on the fourth ventricle. So from the fourth ventricle, we have foramen of Luschika here. This is foramen of Luschika, foramen of Luschika. This is the fourth ventricle. So when CSF comes into the fourth ventricle, it has some options. The first option, it gets reabsorbed back into the subarachnoid space by the arachnoid granulations, or it can trickle down into the central canal of the spinal cord through another foramen here, which we refer to as the foramen of Magendie. Foramen of Magendie, written like this. So that is how it ends up circulating. We can now look at the spinal cord itself. It is just a thin tubular structure. It is made up of the nervous tissue. It extends from the medulla oblongata, from the brainstem up there, and it goes all the way to the lumbar region of the vertebral column. That is between L1 to L2, that is in the adult people. So this one just shows a cross section of the spinal cord, with also one of the functions of the spinal cord is to coordinate what we refer to as simple reflexes. So the central nervous system, that is the brain and the spinal cord, they control reflexes. The simple reflex is normally controlled by the spinal cord, while the complex reflex is controlled by the brain. So this one is showing us a simple reflex on what happens. For example, this is the skin. When you get pricked, there are structures on the surface of your skin, that is the cutaneous region, that we call receptors. So they'll pick the pain stimuli and then send it through the neuron, that is the nerve, all the way to the central nervous system, that is the spinal cord. And here it will meet another neuron, which is referred to as the interneuron, and the spinal cord will initiate a communication through this nerve here, we call it the motor nerve, to instruct the muscle of the finger to remove the finger from pain. That is why when we prick you, if you get pricked, even when you are looking the other side, you will remove your finger first before you check to see what is pricking you. So the function of simple reflex is to actually remove the body from damage as quickly as possible before the brain interprets what is the cause of the pain and instructs you whether to run away or to move away from the stimuli. Therefore, there will be two communications when information reaches the spinal cord. The first information will take this road through the interneuron to remove the finger first, and then another information will ascend all the way to the brain for the brain to analyze and interpret to find out what is causing the pain, and if it is still harmful to you, for example, if it is a snake, then the brain will tell you you need to run away. So that's just an illustration on how the brain, that is the central nervous system, coordinates some of these information. This is still an illustration of what we have just said. There is a prick here. Information is picked, is taken to the spinal cord. Before it goes to the brain, a quick reflex is done, and quick information is taken back to the muscles of the finger to remove the finger from the pain before another information is sent to the brain for further action of what is happening there. These are just the 12 cranial nerves that we had already mentioned. The first cranial nerve is called olfactory. It's a pure sensory nerve. It deals with the sense of smell. The second cranial nerve is a pure sensory nerve. It's called optic nerve. It deals with the sense of sight, that is seeing. The third cranial nerve is a pure motor nerve. It deals with a number of aspects, especially also in the eyes, the muscles that are involved in the eye movement. We shall see in the subsequent slide. Trochlea is a pure motor nerve. It is cranial nerve number 4. We shall also mention one of its functions on the what we refer to as superior oblique muscles. Then we have cranial nerve number 5. It is a mixed nerve. Mixed meaning it has both sensory and motor. Cranial nerve number 6 is purely motor. Cranial nerve number 7 is mixed. Cranial nerve number 8, which is called vestibulocochlear, is purely sensory. It deals with the issues to do with the ear, hearing and balance. Cranial nerve number 9 is known as gross pharyngeal. It's a mixed nerve. Cranial nerve number 10 is known as Vecher's, is mixed. Cranial nerve number 11 is known as accessory nerve, is purely motor. Mostly it deals with the muscles from around the neck here, this nerve here. Then the cranial nerve number 12 is hypoglossal, is a motor nerve. It has functions on coordination of the tongue. So this structure here is the structure of the eye. To illustrate some of the muscles of the eye, these muscles, they move the eye in different directions. For example, when you want to move your eye towards the nose, that is the medial aspect, the muscle that will do that function is called medial rectus muscle. The medial rectus muscle is supplied by oculomotor nerve, that is nerve, cranial nerve number 3. It's a purely motor nerve. When you want to move your eye towards outside, this side, that is what we refer to as the lateral side, then the muscle that will help you to do that function is known as the lateral rectus muscle. So it moves the eye outwards, and this muscle is supplied by cranial nerve number 6, which is known as the abducens nerve. It is purely motor nerve. We have another muscle that we refer to as the superior rectus muscle. Its function is to move the eye upward. That is upward movement. It's also supplied by oculomotor. There's another muscle down here, it's called inferior rectus muscle. It moves the eye towards downwards. It's also supplied by oculomotor. So those are just to remind you some of the functions or the innervation of the cranial nerves on the recti muscles of the eye and the function they serve. Back to our spinal nerves, there are 31 pairs. In the cervical region, we have 8 of them. In the thoracic region, we have 12. In the lumbar region, we have 5. In the sacral region, we have 5. And in the coccygeal region, we have 1. So the total gives you 31 pairs. Remember, all of them are mixed, meaning they have both sensory and motor aspects. So when we talk of cervical region, just this region around the neck, between the head and the thorax, we call it the cervical. So in that region, we have 8 cervical pairs of nerves. Then the region between the neck and the abdomen is known as the thoracic region. We have 12 pairs of spinal nerves around that area. The area between the thoracic and the pelvic is called the lumbar region. We have 5 lumbar pairs of cranial nerves. Then an area between the pelvic and below the cervix, the coccygeal region is called the sacral region. Then we have 5 pairs of cranial nerves at that region. The last region is the coccygeal region, which is the tail, and at that point we have one pair of the spinal nerve. This is a cross-section of the spinal cord. At the center, we have what we refer to as the central canal. Then this is the nerve. Remember, we said all spinal nerves are mixed nerves, meaning they have both sensory and motor aspects. So before we come out as one proper nerve, you need to know that at this point, this is called the dorsal root ganglion, and this is the point where the sensory aspect of this nerve joins the spinal cord, while the ventral root is the point where the motor component of this nerve joins this spinal nerve. So you can see you have a component of sensory and a component of motor. So they come together to give us one spinal nerve. That is why we said all spinal nerves are actually mixed, meaning they have both sensory and motor. So that is basically the cross-section of the spinal cord and the arrangement of those nerves. Remember, the center we have the central canal, and in this center we have CSF that we produced up there from the brain. After now trickling through what we refer to as the foramen of Magendie, it gets into the central canal of the spinal cord, all the way down there. This is still a repetition of the coordination, what happens when you get pricked, information is taken to the central nervous system, that is the spinal cord. A symbol reflex is done, then quick information is sent back to the muscle, instructing the hand to remove, after the muscle to remove the hand from the stimuli. So that gives the first response, and then information is still sent again to the brain, now for fine interpretation and further action to what is taking place at that point. So that is still the continuation of what we have already explained. So let's look at the sympathetic and parasympathetic systems. These are the components of the autonomic nervous system. Autonomic nervous system innervates visceral organs. It's very important to know that autonomic nervous system innervates involuntary muscles. So it innervates involuntary muscles in the visceral organs. Visceral organs, example, are organs that are found in the abdomen. Example is the kidney. Another example can be the liver. Another example is the heart. The muscles on these organs are involuntary. meaning you have no control over them. For example, if we talk about the heart, we know the function of the heart is to pump blood all the way from the day you are born up to now. There is no single day you can wake up and instruct the heart to pump, instead of pumping, maybe let's say 72 beats per minute, you wake up one morning and tell the heart, hey, Mr. Heart, today I want you to pump at the speed of 98 beats per minute. That is not possible. You have no control over that. The only thing that controls the involuntary aspects is the autonomic nervous system. That is an example. Another issue, for example, is the respiratory rate. The other issues is all about to do with smooth muscles in the eyes. For example, in the eye, we have autonomic nervous system, particularly the sympathetic system, which innervates the muscles we refer to as dilator pupillae, to dilate the pupil. When you are facing danger, remember autonomic nervous system, the two components, which is the sympathetic and the parasympathetic, it's important to know that the sympathetic controls or prepares the body for two things, that is fight or flight. That is to face the enemy and fight, or just take off from the enemy if the threat is beyond your reach. So sympathetic component is normally activated when there is stress to prepare the body to fight or to run away, what we are referring as flight. And this aspect also explains why when patients die, their pupils are dilated. It is because during death, death is a frightening aspect, and when you are frightened, you are under sympathetic state. So whenever the sympathetic state is stimulated, one of the areas it innervates is the eye, particularly the pupil, the muscle we call dilator pupillae. Therefore, whenever the sympathetic division is activated, it will act on the dilator pupillae muscles to dilate the pupil. The parasympathetic system functions just to relax and digest. It has no issues. It also innervates the visceral structures and smooth muscles. What you need to know is that these two divisions, that is the sympathetic and the parasympathetic, they are antagonistic. They are antagonistic, meaning they do the opposite of what the other does. For example, if the role of sympathetic division on the eye is to dilate the eye, the parasympathetic does totally an opposite function on it. Instead of dilating the eye, it constricts the eye. For example, when it comes to the lungs, the sympathetic division will dilate the bronchi. But when a patient or an individual is in the parasympathetic system or division, then instead of dilating the bronchi, the parasympathetics actually constrict the bronchi. So these are just illustrations, and it's important to remember these two divisions of autonomic nervous system are antagonistic. They antagonize one another, meaning one division opposes the other. One good thing is that all of them cannot be in action at the same time. Sometimes, in most cases, you are in one state at a time. So you can be in a sympathetic state, for example, when you are facing danger, and you can also be in a parasympathetic state when you are free, relaxed, and there is no danger. These are just the structures, the visceral structures that are actually innervated by the autonomic nervous system. More of a repetition of what we have explained. For example, the sympathetic system innervates the eye and it dilates the eye. So when you are in a sympathetic state, your eyes will be dilated so as to let more light into the eye and see clearly where you can run through or how you can deal with that problem. Also, what you need to remember, when sympathetic system is activated, then it can inhibit salivation. In the lungs, sympathetic system relaxes the bronchi. In the heart, the sympathetic system will accelerate the heartbeat. For example, when you are anxious or you are nervous or you are afraid of anything, then you are in the sympathetic state. Therefore, your heart will beat so fast because of this sympathetic division. That's why we are saying that on the heart, the sympathetic division actually accelerates the heartbeat. When it comes in the stomach, it will inhibit what we call peristalsis. In the liver, the sympathetic state will stimulate production of glucose to give you more energy. Remember, you are in a frightening state. So you need more energy so that you can tackle the problem. So the sympathetic will stimulate the liver to produce glucose. In the kidney, it will cause secretion of adrenaline, and it can inhibit contraction of the renal bladder. Parasympathetic does quite different from what the sympathetic does. For example, instead of dilating the pupils, it constricts the pupils. For example, when we talk of the bronchi, when sympathetic is actually relaxing the bronchi, the parasympathetic is constricting the bronchi, and so on and so forth. So what is a neuron? This is another aspect of the nervous system that we need to look at. A neuron is actually a basic functional unit of the nervous system. Some people confuse a neuron and a nerve. The neuron is the basic unit. Therefore, groups of neurons will give you a nerve. And these neurons can be classified on several aspects. For example, when we look at the direction of the nerve, where the information is being collected, then we can have afferent nerves, efferent nerves, and interneurons. Afferent nerves are just sensory nerves. They are ascending. They take information from the surface, then takes it to the brain or the central nervous system. So that's why we call it ascending pathway. All sensory neurons, they take information to the brain for interpretation. While all the motor neurons, they take information from the brain towards the structure. It could be the muscle, it could be the organ, to take action. So that's why we are saying the nerves that take information to the central nervous system, that is the brain and the spinal cord, are known as sensory nerves. And that pathway is also called the afferent. While the nerves that take information from the central nervous system toward the organs Those are what we refer to as the efferent nerves, and there are two. You can have the upper neuron and the lower neuron. Remember, in the afferent ascending order, that is the sensory pathway, we have three nerves. So the ascending pathway is a pathway of three neurons, what we refer to as the first order, second order, third order. While the afferent or descending pathway, which is the motor pathway, has only two segments of nerves in that pathway. That is what we refer to as the upper neuron and the lower neuron. The upper neuron is confined in the central nervous system. That is from the brain and the spinal cord. So the nerve within the central nervous system, which is the motor aspect, is what we refer to as the upper neuron. While the neuron that leaves the spine, the spinal cord, towards the organ or to the surface of the muscle is what we refer to as the lower motor neuron. Just to mention a pathology, example of what happens when there's a lesion on the upper neuron, it means the neuron, the motor neuron within the central nervous system has a lesion. The presentation will be what we refer to as spastic paralysis. Spastic paralysis. While if you have a lesion on the lower motor neuron, that is the neuron that is from the spinal cord towards the effector muscle. When you have a lesion on that neuron, what we are calling the lower motor neuron, then the patient will present with what we refer to as flaccid paralysis. That is one of the easiest way to tell whether your patient is either having an upper motor neuron lesion or a lower motor neuron lesion. The nerves can also be classified based on the morphology. And in this category, we have unipolar, pseudopolar, bipolar, and multipolar. We shall explain it in the subsequent slide. You can also classify neurons based on the type of modalities. That is what we have referred to as sensory and motor. We have already mentioned that previously. So this is how a nerve looks like. This is the nucleus. This big thing you see here, we call it the soma of the cell body. Cell body. And then you can see there are dendrites here. These spikes you see here are what we refer to as the spikes. This long structure here is the axon. In between you can see there is what we call the Schwann cells. And these internodes are what we call the nodes of ganglia. And what is covering the axon is the myelin sheath. At the end of the axon, we have projections we refer to as axonal terminals. So the dendrites, they take information to the cell body, what we call the soma, while the axon takes information from the cell body towards the central nervous system. So at the dendrites, there are also receptors. Sometimes we call them nerve endings. So there are different types of receptors. They pick the stimuli and then pass it to the dendrites. The dendrites will take it to the cell body. That information will be summed up in the cell body. Once it reaches a threshold, then it can be propagated towards the central nervous system. So the information that is summed here after collection is what we call graded potential. So the graded potential cannot be propagated until it reaches what we refer to as a threshold. That is when now it becomes an action potential and it's taken towards the central nervous system. So that is how this neuron works. Remember, at the axonal terminals, we normally have what we call a synaptic node, where this neuron joins or communicates with the other neuron as the lineage continues towards the central nervous system. These are just the classification of neurons based on the morphology. For example, when you have the cell body here, what you are seeing, this circular thing is the cell body. Then this is the unipolar, because there's only one axon from the cell body. When we have the cell body in between, and you have this extension and this extension here, that is what we refer to as bipolar, meaning there are two projections from here and from there. Then we have another pseudo unipolar. The cell body is somehow from this point, it wants to resemble this. That's why we refer to it as pseudo, meaning it's not purely unipolar. Then we have the multipolar, meaning there are several projections coming from the cell body. So this classification depends on how many projections are leaving the cell body. If it is one projection, then that is unipolar. If there are two projections leaving the cell body, then we call it bipolar. But in this point here, it is a pseudopolar because it's a pseudo-unipolar, because apart from one projection leaving the cell body, there is another pseudo projection here. Then when it comes to this, the cell body has got several projections, the dendrites, and that one becomes a multipolar cell. So thank you for attending this presentation. We have tried just to summarize and touch on various issues that are basic and important. But generally, the nervous system is very, very detailed, is very complex. We cannot finish it within one presentation. So next time we'll try to look at various aspects of individual classification as we go deeper on how it works and its relevance in the body. So thank you for attending the presentation. This is our YouTube handle. You can subscribe, you can drop a comment, you can ask a question, you can give an input. So I think we can call it a day. Thank you and have a nice day. Thank you.